we have got a whole bunch of you saying, we want allopatric and sympatric speciation, and what is speciation, and blah, 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 and you're getting all panicky. Just chill, okay? Get your zen, center. Yeah. Okay. Get two types of speciation. Okay, first of all, what's speciation? Speciation is, now let's think hard. The word says species, so speciation, to create a new species. That's what speciation is to create a new species. Now, how would we create a new species? Well, there would be a mutation of some sort, and if the mutation was bad, it would be a lethal mutation and the thing would die, okay? If it was a good mutation, well, hello, it then becomes fixed because that organism continues to live. And sometimes there's a mutation that isn't good or bad, it just sort of sits there and waits until conditions are different. And then, if those conditions, when they become different, if that mutation helps the organism survive, then it becomes a fixed mutation. And if it doesn't and the organism dies, well, hello, it was a lethal mutation. So that's mutations done. Boom, finished. Okay, then we look at allopatric and sympatric speciation. Now, and if you look here, allopatric and sympatric. Now, sympatric starts with an S. So sympatric leads to sexual isolation or reproductive isolation. In other words, sympatric speciation will result in the organisms not being able to mate. Okay? The first thing you would have to do is be near each other. Okay? But with sympatric speciation, the biggest thing is that there's reproductive or sexual isolation. They no longer want to breed with each other. And that would be anything from when the males do their little mating calls and their little mating dances. Like, have you ever seen peacocks?
I have, actually.
It is
the
too
male
cute.
peacocks.
They strut around and their tail feathers go and all the rest of it. But at the end of the day, if the female doesn't want that little guy near her, well, guess what? He doesn't get to do anything.
I like
and
that.
there is no reproduction.
Well, it's good because he's got the pretty feathers, so
exactly.
She has the choice.
But the minute, there are all kinds of little things that could change the sexual isolation, reproductive isolation. Be as simple as the female doesn't like the dance that goes before it. In other words, where he's now trying to woo
dance.
her. There we go. Okay, so that's sympatric. But it doesn't matter what causes it. At the end of the day, it is the reproductive isolation, whereas allopatric, there is one big, huge, major obstacle in the way of the species. So we have a species A and we have a species B. And there is either a huge wall of water, the sea, or it could be a big mountain range, or it could be the Great Canyon down the middle. There is a huge, big physical obstacle that separates these little organisms, so that species A and species or species A and species B parts of the species, they are separated. And after 100 or 200 or 300 or 1,000,000 different thousand years, they now get together. They've all changed with little bits and pieces all the way along, that by the time they try to get back together again, they are so different from each other that there is no way it's going to get together again. All right, and that is what allopatric. So allopatric, there is always one big barrier. This is a physical barrier, and that physical barrier can be a geographical barrier of some sort. So it is a sea or a huge fast-flowing river or the Grand Canyon or whatever, but they are separated, okay, by something that is physical. Whereas sympatric, the females just don't like the way the males do their little dance, or the mating call, or the physical change of the penile structure and the vaginal structure, or the female receptive structure and the male insertion structure. They just just don't work together anymore. And when that happens, we have sympatric speciation.
Kathy,
There we
I
go.
know. I'm
End of story.
gonna like try and like rush us. Henry,
Okay, tips.
Henry wants to know. He says, "What are the tips in our tummy? Well, in your tummy, I've got no
The
tips."
tips in my tummy, that sit right here is, you are, as far as you're all panicking about evolution, you are going to get a question on sympatric versus allopatric speciation. That's why I've done it here. Okay, they'll give you turtles, tortoises, plants, whatever the case is, and they'll say to you, and then there was this big mass of water, there was a sea, there was an ocean, there was a whatever barrier, and the minute there's a barrier, you know it's allopatric. Okay, and that allopatric will lead to sympatric speciation, but at the beginning it is allopatric because there's a barrier. If there is no barrier, it is sympatric. So that's number one. Number two, they'll give you some other primate and a human being, and they'll give you the skulls, and they'll ask you which skull is good and which skull is bad, and how do you know that they're bipedal? Bipedal means with two legs. So how are they upright versus like a
monkey.
Okay. You'll probably get a phylogenetic tree because someone somewhere in a department is hell-bent on phylogenetic trees. They love phylogenetic trees. So what that is just a whole bunch of organisms and how they all join up and develop, so to show we all come from a common ancestor. They will ask you in a multi-choice, possibly, Lamarck's theories. Remember that it is acquired characteristics and use and disuse.
Do you think they'll ask Wallace's theory as well? There was a few questions on that.
Not really, because, I mean, Wallace, you've only got the Wallace line, and that's really not that important. Okay, genetics. As far as genetics are concerned, make sure they will definitely give you some form of genetic question. They may give you a Punnett square that you have to draw, but they would also, they like their pedigree charts. But the kids battle with them, so they may decide not to give you a pedigree chart. If they give you a pedigree chart, you know how to do it now anyway, as well as a Punnett square. You will definitely, without a doubt, I promise you now, you will get something on protein synthesis. They will give you that DNA, and as long as you remember you've got your DNA and your messenger RNA is a codon, and the anticodon, which is the opposite of the codon, will be your transfer RNA, which makes the transfer RNA the same as the messenger RNA. Okay, just if you remember that, we've done it twice this afternoon. Meiosis, they'll ask you to recognize the phases. Interphase, no one can see anything. Okay. Metaphase, the chromosomes are always in the middle. Anaphase, they're pulling towards the top. And prophase, they are going to be all over the cell but not distinguished. They're not attached to any spindle fibers. Okay, so that's prophase. Anaphase, they're pulling towards the poles. Telophase is easy, they're dividing. I'm trying to think quickly. What else is there?
I don't know if this is a tip. We've got quite a few questions also about on how to write essays and how they should be written in for this exam.
Essays are simple. They're 20 marks, okay? What you need to know is 3 of those marks are going to be the way you wrote it. So did you put in paragraphs? Did you put full stops? Did you put commas? And does it make sort of sense? Okay. The rest of your essay, take the question and break that question up into pieces and answer each piece. And if you do that, you follow a logical process, which means you have to get your 3 out of 17, your 3 out of 20. The other 17 marks will be based on what you've answered. Also, people, they are going to give you something on all the different processes within genetics, in other words, genetic engineering. And they probably give you some other fruit and ask your opinion on whether genetic engineering is good or bad. Okay, cloning, you must know what a clone is versus genetic engineering. At the end of the day, people have taken genetic engineering and they make our lives a lot easier because of that genetic engineering. So it has its pros, it has its cons. Sit down and write out just for yourself this evening or when we finish here, 3 or 4 advantages and 3 or 4 disadvantages of genetic engineering. I'm not going to give them to you. I want you to do it because you need to sit down and choose which are the easiest for you to remember. What makes the most sense for you? So just make sure you know that. Out of Africa hypothesis, they asked it in the Gauteng prelim as the essay question. I think it has merit as an essay. And I mean, the same people that sent the Gauteng prelim are not the same people that are setting your final paper was set last August. So it's not the same. So because it was as an essay question, it cannot be possible that it is an essay question at the end of the year. But make sure that you understand what that out of Africa hypothesis was, and also the difference between mitochondrial DNA and your nuclear DNA. So just so that it may just be a 1 or 2 mark question. Think before you ink. Do not start with the short questions. Do not start with the multi-choices. You do the multi-choice questions absolutely last. You do your short questions second last, and you do your long questions first. Start with your long questions so that you don't waste time. And you can always do if you don't write something for your long questions, it's naught. But if you've got to write 3 or 4 words down for your short questions, you can do that in 3 or 4 minutes. Okay, people, I have to wrap up.
Yeah,
I want to very much, Indiana.
Thank you, Kathy, and thank you, Mindsetters, for joining us.
Yeah.
I'm so sorry. We could have never, we could have a whole year and we would never get through everything. We could never have such a long show anyway. But basically, what I want to say is, guys, please just go into the Facebook page. You guys are incredible. We've got Shakira, we've got Nicole, we've got Jimmy, we've got all of these Mindsetters that are actually helping other Mindsetters out. So go and put your questions there. Help other Mindsetters, guys, help each other out, do it. And good luck. You guys are going to do awesome. We believe in you.
Just think before you ink, don't panic, and start with your long questions first.
And listen to
and I'm going to see you on Sunday. So Sunday we're going to tackle paper 2, which is really a lovely paper.
Yay!
Okay, so be good, study hard, good luck for tomorrow.
See you on Sunday.
See you on Sunday. Bye-bye. Bye.